Hi everybody, how are you doing? So we'll continue with this video further down to the molecular base of carcinogenesis part 2. In this video we will be talking about the environmental causes of cancers, which will encompass not only the physical and chemical causes, chemical mutagens, but also some of the viral and bacterial carcinogens which affect the various cancers. So we'll be focusing on environmental viruses and inherited errors in this video, and finally we will close the video with the overview of carcinogenesis, which is based upon colorectal carcinogenesis as given by Vogelstein. As you know that cancers cannot be caused by itself, you need something to poke into the DNA and change it. Those things which actually change the DNA structure are called as mutagens or carcinogens, and they can be wide varieties in nature. For example, in environment we have chemicals which can change or mutate the DNA irreversibly and lead to a permanent damage. Then there are radiations, specifically UV radiations, cosmic radiations, or X-rays, which can also do the same function of damaging the DNA irreversibly and leading to mutations. Gives you the overall view of the carcinogens present in the environment and the occupation to which it is associated, and the cancer sites which are directly linked to the this type of carcinogens. Like for example, lungs are directly linked to the arsenic poisoning and usually happen in electricians, smelters, and many or other stuffs. Similarly, for like benzene, it is related to lymph node and blood cancers, and usually the occupations which can have an exposure to the benzene are petroleum, painting, detergent, and rubber. Like for example, also some HPV viruses, like HPV viruses, which can lead to cervical cancer, or Helicobacter pylori, which is associated with the stomach cancer. Moving to specific topics of how environments lead to certain cancers, we begin with the lung cancer. As you know, lung cancer is one of the most common and serious types of cancer, and it is directly associated with a single factor of environment, that is a cigarette smoking, which usually is responsible for 70% of the cancers. Because tobacco in itself contains around 60 different types of toxic substances which can be carcinogenic, which can lead to mutation in a DNA. Lung cancer is primarily of two main forms. Those are NSC lung cancer and SC lung cancer. Non-small cell lung cancer, which constitutes about 84% of the lung cancers, while 13% of the lung cancers are small cell lung cancers. In Saudi Arabia, it is the fifth most common cancer among Saudi males, whereas the 15th most common cancer in Saudi women, and in worldwide it is usually the second most common cancer in both men and women. Maybe probably it may be now the first one, surpassing the colorectal cancer. I have given you the links also, you can click them to read more. Another type of exposure which can lead to an cancer is the viruses which causes the cervical cancer, is the HPV. HPV is directly linked to the cervical cancer incidences, as well as the cause is one of the most important risk factors of causing cervical cancer. Among 100 different kinds of an HPV infection, type 16 and HPV type 18 are responsible for most cases of cervical cancers. However, HPV can cause also skin warts, genital warts, and other skin disorders. In world, third most common cancer. However, in Saudi Arabia, it is eighth most common cancer among women from the ages of 30 to 44, and 99% of the HPV cervical cancers is the squamous cell cancer of the cervix. Then adenocarcinoma follows the suit. You can see how HPV infection leads to dysplasia and a change in the morphology of the cervix and later the carcinogenesis. There is a link also which you can follow and read more about it. Gastric cancer is also one of the important cancers affecting the worldwide population. It's the second leading reason of mortality among the world, and usually it has many modifiable risks like eating salted, smoked foods, or smoking cigarettes. But the environmental exposure to H. pylori or Epstein-Barr viruses have been linked more often in these cancers. On the right hand side, there is a graphics which gives the mechanism of H. pylori infection, how it leads to the inflammation concomitantly working with the interleukins and TNFs. This inflammation is then, if left untreated, lead to oxidative stress and then low acidity, which coupled with the other kinds of undisturbed mutations of cadherin or catenin can lead to metaplasia and dysplasia, and finally gastric adenocarcinoma. So underlying cause is the chronic infection by H. pylori. In Saudi Arabia, usually the incidence is 2.7% among newly diagnosed cases, and it's ranked 12th among women and mesothelioma is another cancer which is directly linked with the exposure to the carcinogen called as asbestos. Usually, the people which are in construction business or in navy are susceptible to it. It is aggressive form of cancer which affects the linings of lungs, abdomen, and heart, and usually the expectancy is 12 to 21 months after the diagnosis. The asbestos fiber lodge in the lining of lungs or heart or abdomen and cause the cellular changes, dysplasia, and finally it leads to germline mutations in BRCA gene, which are associated with the high incidence of mesothelioma. One of the most extensively studied cancer is colorectal cancer, and it starts in colon and rectum, and usually depending upon the types of the mutations and which genes and what is mutated, it is categorized into three different types like chromosomal instability, microsatellite instability, and CIMP island methylator phenotype. 80 to 85% of all these CRCs belong to SYN category, where the mutations happen in either oncogenes or tumor suppressor genes or both. Then there is a microsatellite instability, where the mutations usually happen in one wing of genes, that is mismatch repair genes, and they constitute the microsatellite regions and MLH and MSH 2 to 6 genes. All these are repair genes which when affected can lead to MSI type of colorectal cancer. Then third category is now classified as the one which happened because of the gene silencing. The gene silencing is another type of a mutation where the gene in itself is not synthesizing any viable protein because of the mutations upstream, and usually they constitute about 35% of the heredity cancers in colorectal cancer. In Saudi Arabia, it ranks first in men and third in women, with the occurrence rate of 9.6 to 100,000, usually present in the younger age more often than the older one. I have given you the links which you can read later on. Retinoblastoma is one of the important cancers which comes under inherited defects of cancers and usually affects the eyes in early childhood below the age of 5. The affected gene is a tumor suppressor gene called as retinoblastoma RB1 gene. Since it is a tumor suppressor gene, it follows the recessive model of inheritance, and you need two hits to cause the defect in phenotype. Usually the deletions of the chromosome 13 is responsible. Deletions of chromosome 13 affects the zygote first, And you will see in the diagram that if there is a single mutation, then the progeny will be the carriers, which will carry on further until there is a second mutation which affects both the genes, and it will be reflected as cancer in the mutant form. So you need two hits to happen in the retinoblastoma gene to be able to cause cancer. Usually hereditary, one-third of all the retinoblastomas are the hereditary cancers. Another inherited defect is the translocation in Burkitt's lymphoma. You remember that translocations are the gene mutations in which two chromosomes are involved. They exchange their genetic material in reciprocation. In Burkitt's lymphoma, what happens is that there is a translocation in the MYC gene from chromosome 8 to 14. They exchange the material. The MYC oncogene goes and fuses with the IgG gene. And as you remember, I told you that the genetic effect in the gross mutations is because of the unnatural fused protein. Here the fused protein is between the IgG and MYC gene, which behaves like an aggressive oncogene. And this MYC gene is involved in the cellular division, and usually in B cell lineage, it causes an expression and exponential growth of the B cell in and causes non-Hodgkin's lymphoma. Another similar example is the CML, where the translocation happens between chromosome 9 and chromosome 22, and leads to the fusion of the genes which code for the another unnatural protein called as BCR-ABL fusion protein, and it causes the activation of growth factors and signaling pathways, leading to an proliferative growth, which is then characterized as chronic myeloid leukemia. So if you go the model of carcinogenesis, we know that the development of cancer is a multi-step process in which normal cells gradually progress to malignancy, and it is impossible for a single mutations to cause cancer. Usually what happens in the cancers is usually multiple mutations in multiple genes are needed to cause the transformation of a normal cell into the malignant one. You will see usually the old age is one of the risk factors for the cancers. You will see most of the cancers affecting the old age because of the accumulation of the mutations, because mutations get opportunities to build up. The complete sequence or the exact sequence of events which are needed for the human cancers to develop is still not completely known, but it is known. What is known is that both arms of the genes, that the oncogenes as well as the tumor suppressor gene, need to be mutated, and mutations in tumor suppressor genes lead to inactivation, and mutations in oncogenes lead to activation. Both inactivation and activation drives the critical steps of tumor initiation and progression. One of the best model of carcinogenesis was given by Bert Vogelstein in around 1988, when he gave the model of carcinogenesis for colorectal cancer. As I have already told you, that it is one of the most important cancers and well-studied cancers in the genetic world. What he has given is that there is a sequence of mutations in different kinds of genes which drive the pathway from the normal to the metastasis. First and foremost, there is an inactivation of the APC gene, which is in the early event of tumor development. Then it follows the mutations in RAS-K or RAS-H, and the final steps are the mutations in SMAD, R2, and TP53. This is particularly true for colorectal cancer as well as gastric cancer. This is the model which he had given from the normal cell to the metastasis. There are certain steps, and each step is followed by certain kinds of mutation in different types of genes. In colorectal cancer, it is the APC which is the foundation mutation. which then follows with the KRAS or BRAF mutation, then SMAD, and finally the BAX, which develops the cancer from large adenoma. This particular sequence of events are called as genetic instability. I have already told you in the colorectal cancer slide that this is the genomic instability or SIN pathway, chromosomal instability pathway. However, there are another different kinds of models which are given right now and are updated on daily basis rather. This is the updated model of carcinogenesis for colorectal cancer, which has been updated lately and is followed now. You can see in the middle there is a chromosomal instability part, which was given originally by Vogelstein, and up being up is the another pathway which is called as micro instability pathway, MIN pathway, which affects usually the DNA repair gene mechanisms. And downwards you can see there is another different low penetrant genes which can drive the same kind of effect, leading to conversion of a normal epithelium to the cancers by different kinds of mechanisms. This is pretty much it for the cancer biology and genetics and the mutations. You can refer to these books for further reading about cancers, particularly the cancer biology by Raymond Rudon. This is one of the best books on cancer. And in this world of coronavirus, take care of yourself and follow these do's and don't nots. I hope you enjoyed the lecture, and if there are certain questions, feel free to ask. Okay, thank you so much. Have a nice day.